Bart is a virologist, is a lead molecular biologist in the Netherlands Institute for Vector, Invasive Plants and Plant Health. So that's what is in short called NIVIP now. And so Bart is also a member of the panel on diagnostic and quality assurance, and he is always presenting new approaches, new for validation, and that's really great to have you in the panel to show all these advances that are made. And but there's something I'm sure a lot of you know, that Bart is also a very creative guy. He must have a hidden twin or something like that, because he's doing so much, like playing in a band, developing board games, etc., on plant health, to disseminate plant health. So I think it's important. That's you. So, but he's also so active in the lab. So, floor is yours.
Thank you very much, Françoise, for this very lovely introduction, and also thank you for the opportunity to talk a little bit about our experiences working with high throughput sequencing and working from different, can you hear me by the way? Okay, yeah. So working from different research applications towards standardized tests and accreditation of high throughput sequencing tests in a diagnostic setting for regulatory plant health. So we've been hearing quite a lot of different approaches already this morning, different types of pipelines. And that's a little bit the daunting thing that is upon us, upon all of us, when venturing into high throughput sequencing. And basically, going towards accreditation is all about making choices. And these are the choices that we've made in our lab. Those are not necessarily the only choices that you can make. You'll notice there's quite some overlap with some of the presentations that we've seen before. But yeah, that's that. So let's dig in. So NIVIP, the Netherlands Institute for Vectors, Invasive Plant Species and Plant Health. So we are a lab associated with the Dutch NPPO in Wageningen. We have approximately 120 staff members working on 7 different organism-specific groups, so bacteriology, virology, entomology, and so on. We also have one molecular group tending to the molecular needs of all those organism-specific groups. So we have diagnostic activities and applied research and all these other cool things that we're doing. So I'm from the molecular group, and just to give you a little bit of an idea, so we have one molecular group for all diagnostic and research questions coming from the entire department, and that's quite diverse. So we have insects and viruses and nematodes and insects again, and well, loads and loads of different types of material, different matrices, different research questions. So we have 8 different researchers who are in direct link with those organism-specific groups, tending to their specific needs. And we are a group driven with cool new toys. And unfortunately, we have the opportunity to test and play with all these new toys and also to implement them. So for us, we've been grouping the different techniques under 4 methods that are also the basis for our accreditation in the flexible scope. So we have conventional PCR applications, real-time PCR applications, Sanger sequencing, and also high throughput sequencing. So, so these are the newest toys that we have in the lab. Like this. So up. Yeah, pointing to myself, obviously. So this is our Hamilton library preparation robot, and these are actually 2 Illumina sequencers that we have in our lab. So coming down to choices, so this is one of the big choices you have to make, which sequencing technique. And to us, Illumina sequencing is the technique that we are most comfortable working with and that works best in our hands. So yeah, choices. First important choice. So, and our molecular tests are part of a diagnostic process. So that's also a very important thing to consider because it's all about context, especially so with high throughput sequencing applications. So we have a sample coming into the lab. It always first goes to an organism-specific group that has a first set of tests performed. And then when there's a need, it can be passed on to our group for molecular testing. And it could be specific endpoint PCRs, real-time PCRs, or sequencing applications. And so all those four methods I mentioned are actually part of our flexible scope, or ISO 17025 flexible scope. And for the rest of the talk on how we went to accreditation, it's important for you to more or less get a little bit of an idea how our flexible scope works. Because the scope that we have agreed with our accreditation body has been essential to be able to do this. And I am aware that for some countries, this level of flexibility is not necessarily possible or very difficult to achieve with the national accreditation bodies. So it's also good to realize that EPPO is in contact with the European Board of Accreditation, trying to promote this level of flexibility for plant health labs, particularly with some regulations for particularly EU countries, with the need to be accredited for all activities. So our phytosanitary flexible scope. Important here is it are not the individual tests that are focus of the scope, and also not a method-based approach for to ensure flexibility that can vary on a method level, for instance. So for us, it's rather the diagnostic process that is the object of the scope for a given pest. And to show you, this is basically so one of the scopes that we have. This is the scope for virology. And you can see that there's a mention of molecular stuff there. So we're also part of the virological scope. So we have these scopes for nematodes, for insects, invasive plants, and so on. So this really very generically lists what it is we test, plant material, and the methods that we can apply. So that's on a method level. So that doesn't really indicate any specific tests, which are, of course, an application of a method. So, but only on a method level. And then it refers to some internal document. And in this internal document, we have specified, it's maybe difficult to read, but so for instance, for tomato brown rugose fruit virus, the matrices, leaves and seeds, and then the different methods that we have validated for that particular pest matrix combination. And then again, further going into the details, really going to the validation reports, or to demonstrate that actually the tests, and that's where we arrive at test level, that the tests that we are using in these green boxes are actually fit for purpose. So it's all there, and really it's a very flexible system to keep on adding validation data, to keep on adding tests on under existing methods, and to slowly but surely build up the entire dossier on accredited pests, or pests that can be diagnosed under accreditation. So high throughput sequencing, already we've heard it quite a lot, and for us that means Illumina sequencing. So since 2014 we've been playing around with these exciting new tools, and mainly in an applied research setting. So track and tracing in outbreak scenarios, or in support of the description of new species, and always working with data sets and trying to extract from those data sets that comprise entire genomes or entire metagenomes, to really get the sequences that are meaningful to us and that we can use to further come towards a conclusion. So these usually were very ad hoc pipelines. Each and every time a slightly different pipeline, slightly different parameters, as Sebastian also mentioned. So you have all these things that are very important and really influence the output of your pipeline, but also constraining a test, so your test results. So with many manual steps, with little standardization. And since 2019, we started to think, well, okay, there's a couple of things that we seem to be doing over and over again, and it might be time to start to work towards standardization, to really describe it and to fix some of these parameters, to fix some of those procedures, to go to diagnostics. But before we were there in 2019, of course, a small word on implementing all this, because we need to have many aspects actually covered before we can really perform these HTS tests. And it's stuff on data analysis, where do you analyze the data, the bioinformatics skills, the backup and the data transfer, what is a test, what are controls, training staff. So in a sense, it's not that different of implementing other methods in your lab, only there are so many aspects to cover, that makes it quite difficult. What makes it even more difficult, it's almost like a kinetic mobile, like this. If you make one of these shapes bigger and larger, the entire thing gets in a disbalance, and you need to start adjusting on somewhere else, which will eventually lead to adjusting in another place. And not to scare you, but as an example, this was our bioinformatics team and our data analysis environment in 2017 or something. We had one guy with one computer doing it. He was a magician. He could do things on Linux nobody ever seen before, and he was like magic. But slowly but surely, you start to work with the tools, also with these graphical softwares like CLC, Genomic Workbench, and then you get multiple users all of a sudden that want to use it. Okay, that's fair. But then we need to have a kind of a local network with licensed servers, and those licensed servers run on Linux, and fortunately we had Lucas to implement all that. But so and then you're fine for a moment, and then you get in more samples, and your servers are too slow. So what next? So move everything to a cloud environment with multiple virtual PCs, and and then we also had to transfer data from the lab to the cloud. The data connection is too slow. What's next? And it's loads and loads of data. So let's ask some guys to have our own dedicated data line. So we have, and we're from the ministry, and probably people from ministry organizations can relate. It's not always that easy to get something done on an IT level with IT security and so on. So, and here we had a couple of guys saying, hey, we have a group of 10 people in an organization of 3,000, and they want to have an own data line. We got it. And then we start to find that, okay, we're using CLC Genomic Workbench together with Genius, but not all the tools in CLC fit our needs. We want more. So we need to create our own custom scripts and to start to do version control of that and so on and so on. Okay, then come in more samples because we have something that works well. And again, we have to increase the scale of data storage, train the new staff to analyze all those samples. then we want the turnover time to be reduced because it's incredibly powerful, but we want it faster. And this is where we are now. And this will be a continuing story. And that's okay. It seems daunting, but you can really identify stops. And you can say, okay, what we have now is fit for purpose. Okay, of course we want to increase, and of course we want to improve. And you've got these bioinformaticians on the tip of their seat waiting to improve. And you've got to slow them down and to say, okay, we can change everything, but what we have now is okay. And don't change what's not broken. So with all that in place, it was time for us to define tests. And using two EPPO documents actually to help us there. So what is a test? A test is a specific application of a method with a test scope, and it includes everything from the nucleic extraction to the data interpretation. And we have two tests defined in our lab. And for these tests, we have really broken it down to 7 main steps. And this is actually quite similar to the EPPO standard PM7151, only their controls are defined as a separate step. For us, it's more of an integrated thing. And so really it goes from sampling, ensuring backup samples, to repeat stuff when needed, RNA extraction, the library preparation, the sequencing, the bioinformatic pipeline. And what I think is really important to stress, your bioinformatic pipeline will produce output, but then really visually assessing all the output, what is needed to ensure that sequences are reliable, that you know, okay, from this incredible bunch of sequences, these are the sequences I'll continue with to draw a conclusion in the context of my diagnostic sample, and then analyze those, report those, and then you're done. So this is for us the entire test. So actually, similar to, for instance, a real-time PCR, if you change any of these change things in these blocks that significantly change the output of your result at the end, then you're talking about a different test. And that's something that we really have to realize. So we could use this maybe and change the sequencing technique. Okay, fair enough. But then it's a different test. And you would need to ensure that the rest would be fit for purpose still, with verification or validation actually. But this is one of the tests that we have for virus detection in host matrices, based on complete viral genomes. And we have another one which is for insects and nematode identification based on complete mitochondrial genomes and ribosomal DNA sequences. So, and again, the same 7 steps apply, only the content of one of the steps is different. But basically and this is where these choices come together. So what it is that you really put together. So these are our tests. And then the next step, of course, is describing and standardizing everything in standard operating procedures. So we have procedures for steps 1 and 2. Steps 3 and 4 are described in standard operating procedures. The bioinformatic pipeline, the interpretation, the selection, everything. It's a forest of documents, and it's a good thing that it's digital, because otherwise, at least we would need to have an entire forest cut down to produce all the paper. Okay, so now we have a test description, and we want to validate. So the question of why to validate no longer really applies. Of course, we all know why to validate, to show that a test is fit for purpose. And that is dependent on the scope for which you use your test. And how do you validate or verify a test? And actually what we noticed is that for these high throughput sequencing tests, we simply can use PM798 as any other test. Only the tough thing was to define the test with the controls and to ensure that it's run reliably. And so you're using, of course, well-characterized biological materials for that. And then it is the standard operating procedures with the material under that scope that you use to validate. And you can go to the lab, start working, and generate all the outputs. At which time you can conclude whether or not a test is fit for purpose or not. So what about subcontracting? Because we were explicitly asked to comment on that. Because part of this methodology, or part of this test, is actually outsourced. So we have the library preparation and the Illumina sequencing now is all done by a commercial company. And so this company is ISO 17025 accredited. So that's a good thing. That's why it fits nicely in under our quality assurance scheme. So the library prep and the sequencing performed is all done with validated protocols. We have agreements with this sequence provider on data yield and data quality. Also agreements on not to multiplex our samples with other customers. And we get all, apart from the sequence data, we get all the quality control data to assess if sequence runs have been performed correctly, so that we really can identify if this is data that we can reliably use in our test. So also we have a certain agreement with them that in case of high priority samples, we can quickly rush it to the lab, to Leiden, and they'll sequence it within a fair amount of time. But I must say, we're currently validating the in-house sequencing, and that will really dramatically speed up the entire process. So another way is this alien control, which is a really important one for us to assure that also the outsourced part of the test is done reliably. So for us, the alien control is slightly, more or less similar to the control that Natasha showed earlier. Only for us, it's healthy Nicotiana benthamiana spiked with these ERCCs as well, and they serve multiple purposes and also for different steps in the entire test. So they serve, as also as Sebastian mentioned, they serve as a positive control or a negative control depending on how you look at it. But these controls are really, really essential in these tests. And maybe some of you are thinking, again, a virological example. Yeah, that's true. But actually, the alien control that we have for the insects and the nematodes is cat DNA. Because there we are focusing on complete mitochondrial genomes and complete ribosomal DNAs. But cats have them. It is a little bit of a word joke. Look what the cat dragged in. That's why we chose cat DNA. Unfortunately, we had to fire all cat lovers from the lab because otherwise we could have some contamination from another source. But yeah, that's life. So at some point you have the facilities and the resources, the software, the test description, the validation data, the proficient staff, and the diagnostic samples coming into the lab. And then you have everything to present to your accreditation body. And yeah, so for us, it took 8 years to assemble all those elements. And as of August of last year, we passed the external audit and are now allowed to perform these 2 tests under accreditation. And I'm very, very confident that labs implementing HTS tests don't have to go all the problem of all those 8 years, and it's much more feasible now. And we would be happy to get in touch to at least show you how we did it. And of course, the added value of this flexible scope is that now we can fairly easily define new tests and add them under the scope. So we have a test currently under development in support of identification of fungal species using pure cultures, and we have one for invasive plant species. So basically every occasion where we would go into the lab and produce more than one PCR product for Sanger sequencing, nowadays we say we don't bother anymore, rather have the HTS data set and solve it like that. To sum up and conclude, so HTS, very powerful tool in plant health diagnostics, but ask yourself, do I really need it? Implementing the method is similar to implementing other methods, but many aspects need to be covered. The concepts for method and test and scope and validation all apply for HTS tests, such as for other tests. And for us, it was able to become accredited even when we outsourced some of that parts of that test. And what was really important for us were the discussions that we had with both the sequence provider and the Dutch accreditation board to really align everything and to convince them that this was all, the proper assurance could be given that those test results are reliable. So thank you for your attention, and I would like to acknowledge the contribution of all these people, but actually many, many more, and I would be happy to answer any of your questions.
Thank you, Bart. And this is really good to see the success you have in NIVIP with accreditation, because when all the discussion started on HTS, there was always, but accreditation is going to be very, very challenging. And so you showed. And what I like in your one of your last words is to say, we have had lots of discussion with our accreditation body. And I hear sometimes people from different meetings being kind of not afraid, but not daring to have this discussion with the accreditation body. And I want to insist that even at EA level, which you mentioned as well, they say discuss with your accreditation body. If the accreditation body says, no, it's not possible, just take it for granted. Just don't take it for granted, just push them, because they may not be very happy first, but that's also what they have to do. They have to discuss with the labs they are accrediting. So thanks for all this, for showing your experience. So I'm sure there are lots of questions. Natasha.
So thank you, Bart. Thank you. Actually, I had a question regarding validation. And if you look just for the test for the viruses, on how many combination virus matrix you did this full validation?
Yeah, so we based the validation on basically tomato brown rugose fruit virus, really as a case, as an example, and then so demonstrated very extensively that it's fit for purpose for this particular model. And then using that, the approach that we have, because we, maybe contrary to some other labs, we don't use kind of a predefined reference set. So for us, it's really important that actually So from the bioinformatic pipeline, which also has underlying databases to identify putative viral contigs that are interesting to follow up. But then there's always this very much detail to the visual verification using all resources available to us, and then also reporting back in the context of the sample. So, and actually this methodology allows us to generate new reports for other virus host combinations And if it meets certain criteria, then we can use that as a demonstration that for this combination, the test was fit for purpose and we can add it under our scope. And similar as the philosophy that we've been using for Sanger sequencing is that actually the last report that we've generated represents the most current analysis of that given combination and is only valid till then. If we redo the analysis for the same combination, but in the meantime, some of the public databases have changed or our insights have changed or whatever, then we have to re-evaluate if it's still fit for purpose. So that's maybe a little bit of a dissimilar approach than maybe in your lab, I don't know.
I have two questions in the back, so Helga first.
I have a question regarding this agreement with Use of HTS. Because I wonder, do you define requirements for them, specific requirements, and you have a kind of assigned agreement with them, and what happens if they do not fulfill your defined requirements?
Yeah. So we do have an agreement which is more or less in the form of this European tender that we had to produce, where we set all the requirements for our sequence provider. In the last year, actually, we formalized that again in kind of a written agreement on what the turnover time is, the costs, but also data amount per sample, and data quality. So, and if one of those aspects is not met, and also combining samples, plant samples from other customers, and yada, yada, yada. So it's a couple of points. So if those are not met, then we are contacted. Sometimes there are technical reasons when they cannot meet this. One of the challenging things is, for instance, is that quite often we do not meet the quality, minimum quality criteria to pass that the service provider has set themselves. So we quite often do not meet them, but we are working with diagnostic samples, and sometimes, this is what I have, and I need the sequence data, so good luck. So for those, and that's also in our standard operating procedures, we allow to proceed within the process, and then we have to assure, looking at the data and looking at the context of everything, that still the data set that was generated was fit for purpose. Because generating 2 gigabases of output, that's usually not that much of a problem, because there's always something in it that will be sequenced. And so we do have these agreements, and we also allow to deviate them under certain circumstances, but we do need to assure and to document, of course, that data that we got is still fit for purpose for this particular test.
This is also accepted by the accreditation.
Yeah.
Or it is deviation.
Yeah.
Helga, I think it's quicker to pass the microphone. Oh, no. Okay, there's a way in the back.
Thank you. You mentioned that you are going to implement the library preparation and the doing a sequencing laboratories and institute. And you, this is an automated library preparation using a robot.
Yeah.
So you are going to develop in that way for a quantitative, in terms of quantitative output and also for standardization. And how flexible is this robot for different library preparations or samples and so on? Yeah.
So again, there we will have a combination of doing library preps manual and automated. And so the automated platform really has it's a Hamilton robot, and Hamilton has these validated protocols for specific kits. So you buy them, you install them, you implement them, and there's not really much way to vary it. So for kind of a routine setting, that is okay. So we're basically validating then a given set side by side, manual and automated. And if there's something else that we would like to do, because there's so many options, and if we would like at some point to implement that in routine diagnostics, then you have been changing this bit here, so the library preparation. So you're talking about a new test. So we need new test descriptions and new validation data. And there's a tendency maybe still that high throughput sequencing is one thing that can solve everything. But actually, if you start thinking in terms of tests, it's many tests. Like we have many real times. And going towards description of those tests in EPPO protocols, that is something that is the next exciting challenge to Gerald.
Quick, because we will be running out of time.
Thank you. Thank you for the interesting talk. Could you comment, let's say, for nematodes, how often or how many samples do you test with this high throughput sequencing test, and how many go to, I don't know, fine?
So for nematodes, this would really be considered as kind of an identification test that replaces the Sanger sequencing. And this is performed on single nematodes. So it's really one nematode that is input for the DNA extraction and the rest of the test. Because they are so challenging, because of the little amount of biological material, we fish out 3 single specimens per population for identification. And if there's a need to have additional samples analyzed based on the results, then we could include more. But typically we have 3, and that is 3 single specimens from a population. And of course, that will not cover all the variation within a population. And if you have a kind of a nematode extract with multiple genera and multiple, of course, then, and fortunately, we have a very strong diagnostic taxonomical team in nematology that really identifies, okay, I want, for this particular specimen, I want to have an identification, and that we use as input in the test.
Thank you.
And if you would like to further discuss about that, no problem.
The last question on the back. That's the last one. Well, it depends if you think it's interesting for the whole group.
So a question about this regarding the subcontracting, especially with the subcontractor, you mentioned that there is no multiplexing with plant samples. What about other field? Food safety or human samples? Did you have some contamination?
Well, we found a coronavirus in our virus pipeline. That's also because subcontracting in a sense is a bit of a black box, of course, and you can have all these agreements, and that's why it's really good to have it very close to you. So we do have some assurance with those controls and with the agreements we have, but doing it in-house really allows you to be really on top of things, and then still part of it will be a little bit of a black box.
Thank you, Bart.